RNA sequencing, called RNA-Seq for short, follows a similar process as array-based gene expression profiling. To extract RNA, you may do poly-A tail selection by using poly-T beads for mRNA selection, but there are protocols that do not require poly-A tail selection. After RNA has been isolated, a complementary DNA library is prepared by reverse transcribing the RNA, then fragmenting the complementary DNA to break it into shorter pieces, and then performing size selection. Linkers are then added, notice the blue and red primers on the ends, followed by massive parallel sequencing where the sample is spread across the flow lanes of the solid substrate. Each single spot on the chip's surface is very diluted, so only one fragment is in each location. Again, each single spot on the chip's surface is very diluted, so only one fragment is in each location. A PCR-like process called bridge amplification is then used to build up the number of fragments at each location. Then when sequencing, a single base is added one at a time and an image is taken to determine which base was added at each location based on the fluorescence emitted reading from the 5' to the 3' direction. This results in sequence reads that are 50 to 300 base pairs in length. After getting the reads are matched to the reference genome in a process called alignment. The human reference genome is a consensus sequence made from several people. Most people will roughly have the same sequence. The number of reads aligning to a given gene allows us to infer how much of that specific gene is expressed in the sample. Therefore, this technique is often called next-generation sequencing. Illumina is a company that dominates this market, having developed different machines including HiSeq, NextSeq, and MySeq. Next, please watch the YouTube video illustrating Illumina's sequencing assay.